So the last lecture of the series is hematoxylin and eosin staining. So hematoxylin is a routine dye in histopathological laboratory. For routine diagnosis, the use of hematoxylin along with eosin is by far favored for viewing cellular and tissue structure detailed by histopathologists. Hematoxylin and eosin demonstrate such a broad range of cytoplasmic, nuclear, and extracellular matrix features. Nearly all textbooks use hematoxylin and eosin images for teaching purposes. So first of all, we will discuss about the hematoxylin. Hematoxylin is a natural and basic dye extracted from Hematoxylin campesинum tree and was first introduced by a German anatomist in 1963. In different countries, different names are used for this tree, that is the Palo di Campeche, logwood extract, bluewood, and blackwood. Hematoxylin has poor staining properties and is normally used in conjunction with mordant. In other words, we can say that hematoxylin is an indirect stain. The ripening or oxidation of the hematoxylin before use is a compulsory step. So there are two ways for the ripening or oxidation of hematoxylin. The first one is spontaneous or natural, and the other is chemical oxidation. So spontaneous or natural way is to place the hematoxylin in open air and atmospheric oxygen, then bind with the hematoxylin to oxidize or ripe it. But the problem is it is very time taking, and it takes about 3 to 4 months to fully oxidize the solution. So in the given picture, the hematoxylin tree model is present with the bark of the tree from which we extract the stain. So this picture represents the skeletal formula of hematoxylin. You can see we have a clear-cut benzene attached with a chromophore and oxochrome. So in chemical way of oxidation, we use different types of the chemical, and those chemicals which are used for the ripening of the hematoxylin is known as oxidant. So there are different types of the oxidants like mercuric oxide, potassium permanganate, potassium iodide, and hydrogen peroxides. So this table represents various types of the oxidants and their amount which is used for 1 gram for the ripening of 1 gram of hematoxylin. As we discussed that hematoxylin is a poor stain and it requires mordant to bind with properly with the tissue. So there are different types of the mordants that can be used for hematoxylin staining. For example, divalent or trivalent metallic salt are used, although simple salt of the metal like aluminium, iron, and chromium can also be used, but conventionally alum salts are used for the staining. So we can use potash alum, we can use ammonium alum, and iron alum as a mordant for the hematoxylin staining. So beside these mordants, simple chloride and sulfate salts of aluminum can also be used. The use of alum salt is just a tradition because initially these were used probably due to being less expensive and easy availability. In routine, Harris hematoxylin is used in the hematoxylin staining. Ripening is done by mercuric oxide, while potassium and ammonium alum are used as mordant. This table depicts various ingredients of the hematoxylin stain preparation containing the stain powder, alcohol, the solvent, potassium, ammonium alum, distilled water, mercury chloride, and glacial acetic acid. So what is the significance of the ingredients? Hematoxylin is a stain or powder, and then absolute alcohol is solvent for hematoxylin, alum is mordant, distilled water is solvent for alum, mercuric oxide is ripening agent, and glacial acetic acid enhance the staining capability of the hematoxylin. This is the procedure for the preparation of the hematoxylin stain. Dissolve hematoxylin powder in absolute alcohol while alum and distilled water with gentle heat. Mix both solutions and bring to boil rapidly. Then remove container from flame and add mercuric oxide. Cool the mixture rapidly and add glacial acetic acid. Filter the stain and store in dark brown color bottles. Eosin is commonly used a counter stain along with hematoxylin. It is a synthetic dye and belongs to the xanthine group of stain, and it is acidic in nature. Cytoplasm, muscle fiber, red cell, collagen, and other connective tissue fibers show affinity for eosin. Eosin has an excellent property of staining different tissue components in different shades, ranging from orange to deep red. Structures that stain rapidly with eosin are termed as eosinophilic. So different commercial brands of eosin are available, namely Eosin Y, Eosin B, Eosin S. The commonly used eosin is Eosin Y. It is a tetrabromo derivative of fluorescein. Orange floxacin may also be used as substitute. Eosin is listed as in IARC, International Agency for Research on Cancer, is class 3 carcinogen. So the picture depicts the skeletal formula for eosin. Preparation. So eosin powder, potassium dichromate, 1 gram each in distilled water up to 100 ml. Prepare separately and then mix both solution. Now when the hematoxylin and eosin solutions are ready, we will perform the staining procedure. So we have summarized the whole procedure in this table. So the top of the table you can see reagents, time and rationale mean why we are performing this step. So first of all we use xylene 1 and then xylene 2 for 5 to 10 minutes each, and the purpose of this step is dewaxing or deparaffinization. Then we use absolute alcohol 1 and absolute alcohol 2 for 2 to 3 minutes and 1 to 2 minutes respectively, and this process is known as dexylation. So while our stains are prepared in aqueous solution and the tissue which was embedded with paraffin wax and then section contained the paraffin wax, therefore this stain cannot be directly applied on the slide because wax is not miscible with water. Therefore we must have to remove the wax and xylene to properly stain the tissue. The third step is hydration. So for hydration we use 90% alcohol, 70% alcohol, 50% alcohol, and then running tap water each 1 to 2 minutes. This whole process is known as hydration. Now the tissue is fully hydrated and the hematoxylin is also in aqueous solution. Both are miscible with each other, so we can use this aqueous solution of hematoxylin to stain the tissue. So as you know that the hematoxylin is a basic stain, it will stain the acidic component of the tissue. So nuclei usually are acidic in nature, so hematoxylin will stain the nuclei. That's why this is known as the nuclear stain. The duration is 1 to 2 minutes for progressive and 3 to 5 minutes for regressive hematoxylin staining procedure. After the hematoxylin application, 1% acid water is used only for regressive steps. This is 1 to 2 tips of the slide, and this process is known as differentiation. Then alkaline running tap water, that is 1% lithium carbonate or 1% ammonium water, is used for 2 to 3 minutes. So this step is known as blowing, and blowing actually means neutralizing the acidic solution that we have used in the previous step, that was 1% acid water, to facilitate next staining technique. So next is eosin stain that we apply for 3 to 5 minutes, and eosin is acidic in nature, so it will stain the basic component of the tissue, and cytoplasm is is majorly basic components, so it is also known as the cytoplasmic staining. After the application of the Eosin staining, we use tap water to wash the extra stain for 1 to 2 minutes. Extra stain mean the stain which has not taken part in the staining reaction. Then we use alcohol 70%, 90% and absolute alcohol for 1 to 2 minutes each, and this process is known as dehydration, because now the staining we have performed the staining, and now we have to observe the slide under the microscope. So for the better visibility and to increase the refractive index of the tissue, we use xylene, and xylene is not miscible with water. That's why we remove water, and the process is known as dehydration. So after the dehydration, we put the slide in xylene 1 and xylene 2 for 3 to 5 minutes each, and this process is known as clearing, which also enhance the refractive index or visibility of the tissue under the microscope.